Hello, everyone. Welcome to today's AHA Stroke Bridge webinar series session. Thank you all for being here. My name is Hanson Chen. I am an instructor in the Department of Neurosurgery at Stanford University. Our goal is to bridge basic science, translational research, and clinical perspectives in stroke by bringing together researchers from different disciplines to share ideas and foster collaboration. This webinar is supported by the HA Stroke Journal, and we hope it continue to grow as a platform for connecting researchers, clinicians, and trainees across the stroke community. I would like to thank Rebecca and Erin for helping this program possible. So today, if you have any questions in the session, please feel free to type them in the Q&A, and we'll address them at the end of the webinar. And please know that this session is being recorded. We are very fortunate to have two outstanding speakers with us today, Dr. Marion Buckwalter from Stanford University and Dr. Louis McCullough from UT Health Houston. And let me start by introducing our first speaker, Dr. Buckwalter. Dr. Buckwalter is a professor of neurology and neurological sciences at Stanford University. She is also the co-director of the Stanford Stroke Recovery Program and deputy director of the Ultra Neuroscience Institute. Today, she will talk about the mechanisms of post-stroke cognitive decline and neuroimmune responses after brain injury. Dr. Buckwalter, thank you for joining us. The floor is yours.
Thanks so much, Hanson. Can you hear me okay? And can you see my slides?
Yes.
Thanks for the invitation. And to everybody out there, I am excited to share this work with you. And as Hanson said, I'm very open to collaboration. My email is on here. It's just my name. And we're also recruiting for a postdoctoral fellow. If anybody listening is interested, there should be, feel free to contact me. So post-stroke cognitive decline. I thought I'd talk just a little bit to start with why I'm interested in post-stroke cognitive decline. And this is a great paper, actually, because although there's been a lot of progress in acute stroke treatment, there hasn't been as much. We don't have good therapies for people after stroke. So what these people did is they surveyed over 1,000 people with stroke who are living with stroke and asked them what problems do they have. And here you can see the percent of people experiencing each problem. And although mobility, which is probably what you think of first, is right at the top, you can also see that concentration and memory are in the top four. Then the next thing they did is they asked them, are your needs met? So are there therapies or things that have helped you with your needs? And actually only 25% of the people with a mobility problem still felt like their needs were unmet. But if you look at people that had problems with memory or concentration, it's about 50% if you average those two. So cognition is a huge problem. So what is post-stroke dementia to start with? So having a stroke, it turns out, doubles the risk that somebody will develop dementia in the decades after the stroke. So this is a new diagnosis of dementia. And it's incident dementia, meaning a new diagnosis, and it's after accounting for common risk factors of dementia, which include older age, high blood pressure, diabetes, poor diet, lack of exercise, and use of tobacco. The absolute risk and relative risk depends on age. The average is that the risk of dementia is doubled. And it's been an unclear mechanism with unknown risk factors. So the part of this post-stroke dementia that really interests me is this late risk of dementia. And this graph here is copied from a really important study actually published in our stroke journal in 2004. And what they're looking at here in red is people who didn't have dementia prior to their stroke, but then scored as having dementia on a cognitive screen. And what you can see compared to controls who also didn't have dementia is that the people here in red have a higher percent of dementia. And it's in two phases. One is around the year of the stroke. There's strategic damage to the brain. So if somebody, for example, has a problem with language due to where the stroke is, they might score as having dementia here. However, the lines don't stay parallel. This red line moves away from the blue line. And actually, this risk here is even greater than the risk at the time of the stroke. So by 10 years later, more people have a stroke from some other process, dementia from some other process, than the people who got cognitively impaired by the stroke itself. And importantly, this late risk of cognitive decline does not seem to be impacted by preventing a second stroke. So it's not just having more strokes. And I became really convinced of this actually in the basic science lab. So this is Christian Doyle, who is now a professor at University of Arizona. He was a postdoc in my lab when he discovered and developed this mouse model of post-stroke cognitive decline that sort of mirrors this infarct-induced neurodegeneration, this late risk of dementia in people. And what he found was 1 week after the stroke, this is a mouse cortex or the surface of the brain. You can see that that part is just gone from the stroke, like someone took a bite out of the brain. Um, the mice are actually cognitively normal at this time. This is sensory cortex, so they have trouble feeling things with their whiskers, which is one way mice feel things. And they have normal electrophysiology in their hippocampus, and they can do normally on memory tasks. But then 6 weeks later, 7 weeks after the stroke, they develop atrophy and thinning of the cortex, and they also develop a scar that is filled with lymphocytes. So lymphocytes are adaptive immune cells, just like the ones you develop after a vaccine. In this case, we think they're probably developed against brain antigens, like myelin, for example. And lymphocytes, in this case, the B lymphocytes that make the antibodies, um, actually are responsible for this cognitive decline. When Christian ablated these cells 5 days after stroke, he found that mice did not get the cognitive decline. So that was super exciting because it told us that perhaps we could, if the same thing happens in people, we could get rid of lymphocytes in them or deactivate them, just like in other autoimmune diseases, and be able to improve cognition. However, mice are not people. And so the next thing we did is we looked at brains from people from this very famous study called the Rush Religious Orders and Rush Memory and Aging Project. In this study, people join at age 65 or older and undergo cognitive testing. They have an average of 8 years per participant, and they donate their brains when they die. And what the scientists there did is they selected brains for us from people who had no Alzheimer's disease or a minimal amount of Alzheimer's pathology and did have infarcts in their brain. And we asked the question of whether these two types of lymphocytes, B cells and T cells, were increased in people who had an infarct. And what you see here in green is the controls. There's very few T cells and B cells in the normal brain. And then from people with infarcts, people who had dementia had more B cells in their brains, just like the mice. And they statistically did not have more T cells, but there were more T cells overall with infarcts. This middle group is people with infarcts or strokes in their brains who did not have who didn't have dementia, basically. So there is some similarity between people and mice. However, you can't make an exact comparison. Obviously, people are more complicated. And also, these people are often dying many, many, many years after their strokes. But what you can see is that even though there's more B cells here, it's not like the mice where they all have tons of cells. This is a picture actually from a person, the brown cells or the B cells, who had the most B cells, and they have a lot fewer than the mice. And many of these people actually have a normal number. So what we decided then to do was to take a step back and just ask in an unbiased way, if you take people who've had a stroke in the past and you study their blood, can you find evidence of an immune response or some other response that tells you that they are at risk of cognitive decline? So we started this study called the Stroke Cog study. It's described in this paper here. And for this study, we recruited people that were at least 5 months after their stroke. The median was 10 months after stroke, so nearly a year. And then we followed them yearly. And on average, we had a median of 26 months of follow-up in these people. We then took a drop of their blood and measured over 7,000 proteins in the blood with an aptamer-based method. And what we asked first was, is there any difference in the blood between people who are healthy, who've never had a stroke, and people who'd had a stroke? And we got a lot of data from that, as you can imagine, from 7,000 proteins. But one of the things that really stood out to us was a downregulation of this protein, PDGFB, and its entire pathway. So the way to read this is when the curve is over to the right, there's a severe downregulation of a pathway. So not just PDGFB itself, but all the proteins that respond to it and bind to similar receptors were also decreased in the blood of people with stroke. So the next thing we did was to look at our cognitive data. And this is a graph of the change in cognition. So remember, people are an average of 10 months after stroke. And that's when we took the blood. And we're looking to see after we took the blood, what happened to those people. And people who, if the dot is at 0, it means that year over year, there was no real change in their cognition. If the dot is higher, it means they were improving. And if it's lower, it means they were worsening. And the main thing to worsen was actually processing speed and executive functions. This is like speed of thought, keeping lots of things in your head, was getting worse over time. In many of the people, the whole population was significantly lower year over year. But there were people in there, so we divided them in half. And so the top half we called better cognitive trajectory. And these people are essentially not changing. But the worst category, these are the people who are declining year over year, and they're decreasing by 1 standard deviation a year. And really interestingly, the people who are declining are older. Now, of course, old people don't generally do as well as young people, but these cognitive scores are already normalized for age. So it means there's a vulnerability that comes with age, not just that they're getting worse because of age, but that when you're older and you have a stroke, you're getting worse more. Other than that, consistent with other data, we saw that there was no effect of stroke size or location and no effect on stroke severity. So how big the stroke was or how impactful it was doesn't impact this late change of cognition. And we can talk about that if people are interested. So we just asked, what about the proteins in the blood of the people who are really doing better versus the people doing worse? And really interestingly, the same pathway came up again. So within stroke, the people who are doing better look more like normal people without a stroke, and the people doing worse look more strokey, if you will, and they have more of this loss of this protein. This protein is really interested because it is key for maintaining what we call the blood-brain barrier. So maintaining a barrier between the blood and the brain and keeping things in the blood from getting into the brain. And so we developed this hypothesis that if you have low PDGFB, like the people who are getting worse, that you might have a leaky blood-brain barrier, and it would be loss of these blue cells you see over here called pericytes, because one key role of this protein is to recruit new pericytes to new blood vessels and maintain pericytes on blood vessels that already exist. So we hypothesized, just based on the proteins in the blood, that what we would see was chronic leakage of the blood-brain barrier in patients who have post-stroke dementia, and that it would be due to loss of pericytes. And I'm just keeping an eye on the time here, Hanson, don't worry. The next thing we did was we designed a study, and this is an ongoing study, but I'll show you some data from it. So the purpose of the study is to use a special MRI to look at blood-brain barrier leakage. And so we took patients who are 6 to 12 months after stroke and compared them to controls. And I will tell you this study is still ongoing, but we have enough baseline scans from here to ask the question of, is there actually more blood-brain barrier leakage this late after stroke? So everybody knows there's blood-brain barrier leakage within days after stroke, but is there more leakage 6 to 12 months later? And so obviously I'm here giving you the talk. So the answer is yes, we did see more leakage. And unfortunately, people who have chronic stroke have about twice the whole brain leakage of controls. And now these are controls that are matched for cardiovascular risk. So these are people that already have white matter disease, high blood pressure, diabetes. They're matched for age also with the stroke participants. And it was highly significant. The patterns are also really interesting. There were a number of patients, and there's 2 of them here that are really marked, where this is the acute stroke lesion here in white. And then if you go over past this dotted line, these are scans from 6 to 12 months later in the same people. And this is the leakage scan. So the focus of the leakage is actually right around the stroke in these people. In other people, it's not so obvious. Here's the stroke, and it's really not as obvious. And then other people don't really have a lot of leakage at all late after stroke. However, statistically speaking, it's not just the whole brain. It's even if you take away the stroke lesion and white matter regions that do also have more leakage than white matter lesions and controls, you still have increased leakage in the people with stroke. So there's a whole brain phenomenon. So next we wanted to ask from our hypothesis, basically, are these blue cells actually missing in people that die of dementia? And so the same people that we've stained for the lymphocytes, we now went back to brain sections near the stroke, and we asked, are these blue cells missing as we predicted from the proteomics? So is there a structural deficit in the blood vessel? And so this is what the healthy controls look like. In green here is a blood vessel. You can see a rim here of red, and this red here is the marker we're using to label the pericytes, this is actually the receptor for the protein that's low. And in a normal person, you often see that pretty much all the blood vessels are covered by pericytes, and that maintains the barrier. But when we quantified, what we saw is that stroke was different. So here are the healthy controls. Here are the people with infarcts but no dementia. And here you can see the people with infarcts and dementia. So these are people that had dementia prior to death, and it's the tissue right near their stroke. But what you can see is that the median value is actually close to 0 in these people. And this is sort of what it looks like. So here's a person who died with an infarct and no dementia. Here's the blood vessel. Here are the pericytes, and there's a pretty good match in this particular person. And then here's somebody with infarcts and dementia who has essentially no pericytes visible. So we saw, unlike the B cells, this seemed to be true in nearly everybody who died with an infarct and dementia. So just to sum up, we see this chronic, what we call adaptive inflammation that involves lymphocytes in the brain after stroke. We have a little bit of other evidence that there's an autoimmune response going on, which I don't have time to talk about. More on that maybe in the future. But the proteomics told us that blood-brain barrier leakage was really a big predictor of who was going to go on to decline in our cohort. And we saw the structural deficit in pericytes that we predicted from the proteomics. So this is in 3 separate groups of volunteers, people with stroke who volunteered for the studies. So next question in the remaining few minutes is, what can we do about it? So along came in the lab another fabulous basic scientist. This is Christy Zara, who's just started her lab at University of Minnesota as an assistant professor. And Christy was interested in this molecule VCAM1. VCAM1 is an important molecule in that it goes up on endothelial cells, or the linings of blood vessels, when there's inflammation going on. And it's responsible for helping immune cells get into the brain. But to do that, it actually opens the blood-brain barrier. It binds to a partner on immune cells called VLA4. So this is the interaction. Here's the VCAM1, here's the VLA4. And early on, when Christy came to the lab, we collaborated with another lab that discovered that VCAM1 gets shed into the blood and its levels go up with aging. And actually in aging mice, if you block VCAM, they get smart again. And Christy did these experiments for the other lab. So she did memory tests on mice and was able to show that 3 weeks of blocking VCAM improved the cognition in the mice. So we were really interested in, like, is this a candidate protein that might improve cognition in people? So we went to our proteomics study that I just told you about, and there were a lot of proteins that were different, and it turns out that VCAM was one of them. So it's about 20% higher than normal in the people who have chronic stroke compared to the healthy controls. Each dot here is an individual person. And when you look at these 2 groups of people with the worst cognitive trajectory versus better, it's even higher in the people with worse cognitive trajectory. So it looks like it goes up in people and it predicts what's going to happen. So these ones that are higher went on to do worse over the next 2 years after the blood draw. We then looked in mice to see, is the protein going up and staying up in our mouse model? And this is the same model I told you about earlier. You can see that VCAM goes up, and even 7 weeks after stroke, it's significantly higher than it is in a naive mouse without a stroke. So we went back to Christian's model, now doing behavior, And the behavior that Christy did was Barnes maze. So what happens is you put a mouse on a maze, and it's on this big plexiglass sheet. It's about 3 feet wide, and all these holes are open to the floor. It's sort of like table height, except for this one here that's shown in green, where we call it the target. It has like a little box so the mice can get in a little corner where they like to be. And the first time they go in the maze, it takes them a long time to find the target. But over time, they learn and they go right to it. So we time when we do different trials and when we put the mouse down, do they remember where the hole is? And this is what a mouse that had a sham surgery and got control IgG looks like. Pre-stroke in the training, it learns. So it goes from about 90 seconds to something like, I guess that's about 50 seconds. And then 1 week after stroke, it also can learn where the hole is. And the same 6 weeks after stroke, because it didn't have a stroke, it had a sham surgery. If you do a stroke, now in the gray, these are mice that had a stroke and got control IgG, and they do terribly. So 1 week after the stroke, they can still remember where the hole is. But 6 weeks later, they cannot find the hole. So their times get really poor. They have gotten dementia late after stroke, if you will. So the next thing we did is we blocked both of those proteins, VCAM1 and VLA4. This is the data with VCAM here in red, and I'll just add the VLA4. And now they're performing like the mice that got a sham surgery. So basically all the groups in these time points pre-surgery and 1 week after surgery can do the task. But 6 weeks later, the mice that got these two blocking antibodies can now perform the task just like mice who didn't have a stroke at all. They don't get dementia. Christy also did a second group of experiments, same mice. This is a different memory task, and the same thing happens. Just very quickly, because I want to stay on time, to look at the mechanism of this, Christy actually did single cell sequencing. So she did RNA sequencing. And one of the really interesting things is, although there were fewer lymphocytes in their strokes, the RNA transcription was not really different in the immune cells. But in the endothelial cells, there were huge differences, and a lot of them had to do with maintenance of the blood-brain barrier and angiogenesis, where basically adding these antibodies was tightening up the blood-brain barrier. That's what it looked like. And so we thought, are these antibodies actually recruiting pericytes back? And so now you're looking at a stain of mouse pericytes. And what Christy found is by treating with either VCAM antibodies or VLA4 antibodies, she could restore the pericyte coverage to the vasculature. So that's these graphs here. And you can see it's actually on both sides. It's significant with the VLA4. And so that was pretty exciting. We could also look at blood-brain barrier leakage in the mice because we have sections where we can look for fibrinogen, which is a protein made in the liver that shouldn't normally be in the brain. And what you can see is that the mice that got control antibody have leakage of fibrinogen, but if you treat them with VCAM1 or VLA4, they don't have any blood-brain barrier leakage. So this now leads us to a hypothesis where we think that people who get cognitive decline after stroke are having activation of the endothelium. And it likely is exacerbated by low PDGFB. So they then can't recruit pericytes and they're letting more immune cells into the brain. And when you block this with anti-VCAM1 or anti-VLA4, you can close the blood-brain barrier and prevent cognition from worsening. So we're really excited about this. I will say, I just want to make sure I thank our participants. So we have amazing participants in these studies. They come in year over year to get their cognition checked. Many of them have gone through MRI scans with contrast, and we really appreciate their contributions, which we hope are going to help us prevent post-stroke dementia. This is the stroke center team that helped us recruit these patients. And then these are the researchers that worked on all the data, and of course, all the funders who we appreciate. And then I'll just hold on this slide for a minute. Both of the data I showed you today, both the human paper and the mouse paper, are not published, but they are available in preprint form on bioRxiv. So if you're interested in learning more, you can look for both of them there. So I think with that, I am exactly on time, and I will end now, and I look forward to taking your questions after Dr. McCulloch's talk. Thank you.
Thank you so much, Dr. Buckwalter, for the excellent presentation. I think these are exciting findings. We will save the questions until the end of the session. Now I'll introduce our second speaker, Dr. Louis McCulloch. Dr. McCulloch is the professor and Huffington Chair of Neurology at UT Health Houston. Her talk today will focus on the biological sex differences, metabolism, and immune mechanisms influencing stroke outcomes. But probably, I'm not sure if Dr. McCulloch has made a big change for the topic today. Dr. McCulloch, thank you for joining us today. Please go ahead.
Thank you. And I will also try and stay on time. And I do tend to speak very quickly. So talking with Marion, we decided I'm going to do something a little different. It might be a little choppy because some of this data is fresh off the press. But we have been working on the detrimental role of social isolation in stroke outcomes. And it integrates really well with another project in our lab that's looking at peripheral inflammation, especially inflammation that's kind of in the gut. So I'm going to talk a little bit about that. So, all right. So I always talk about sex differences because that's a big area in our lab. And it's really pertinent to this topic in stroke because we know that women have much worse outcomes after stroke. They are older when they have their first stroke, which also plays a role. But before I talk about sex differences, I just want to set the frame of the difference between sex and gender. So sex is really chromosomal and hormonal influences. But we know that those really can impact our immune system. So it'll be interesting to talk with Marion after to see what kind of sex differences, if any, came out in her studies, because we know that women have a very different immune system than men. Now, there's gender, which is not really applicable to mouse because it is a sociobiological construct that applies to humans only. But when you talk about stroke outcomes, gender becomes very important. Because women have different outcomes after stroke, but a lot of them have to do with the fact that women are often socially isolated. And that's what we're going to talk a little bit about today. So again, we know that women do worse after stroke, but if you look at factors, so if you do unadjusted analysis, women are 35% more likely to be dead after a stroke 1 year. But it fully reverses, and they actually have a survival advantage if you take into account things like age, stroke severity, and social factors, including social isolation. So social isolation is really, really kind of come to the forefront even more so, although people have known for quite some time that social isolation increases the risk of first stroke, it increases the risk of second stroke, and it increases bad outcomes after stroke. Especially with the COVID pandemic, I think this became even more obvious, but there is something very biological about the detrimental effects of isolation or loneliness. And those are two slightly different constructs, and we can talk a little bit about that if we have time, but it has the same detrimental health impact of smoking 15 cigarettes a day. We know that it increases your risk of by 32% of a stroke, and it also increases dementia. So huge cost to the public health system. So why does social isolation do this? And what is the difference between living alone? Because many people live alone and actually don't have an inflammatory or an immune response. You have to feel subjectively lonely for kind of some of the detrimental effects that we see with isolation. We know it's related to increased cortisol, increased inflammation, IL-6, TNF alpha. We found these in our own patients as well. It causes vascular dysfunction. But one of the issues is when you study this in humans, there's a lot of other confounders that can interact with social isolation, meaning, oh, maybe you can't go to the Walgreens and pick up your medication, or maybe you're not eating healthy foods, or maybe you're drinking or smoking. But for isolation or loneliness, it's really specifically detrimental to women. And 52% of women live alone compared to 29% in elderly women. And that's a lot because they're widowed. And that, of course, will impact stroke outcomes, because if you can't get up 3 steps without assistance, you're going to end up going to a nursing facility rather than going home. The detrimental effects of isolation on stroke have been shown in a multitude of studies. So this is pretty rigorous when you see so many studies showing the same effect of the detrimental effects of isolation. This is also true for dementia. This is a relatively recent analysis looking at dementia and isolation, and people who were isolated also had higher rates of dementia and cognitive decline. But like I mentioned, social isolation is a really tough construct. And how do you study it in animals? Because some of it is being alone, and some of it is that subjective loneliness. And there's so many factors that go into it that affect the health outcomes of loneliness and isolation. Because again, if you're lonely, you may also be depressed, and you may have poorer health behaviors. So we decided probably 15 years ago to try and model this in mice. You can't really model loneliness because, you can't really assess if mice are lonely. So we did just isolation. This is our stroke model. You saw from Marion, she uses a smaller distal cortical infarct. This is in a standard reperfusion model where we put a suture into the internal carotid up into the middle cerebral artery. And I show you the coronal sections here, and you can see the area of white is the area of infarct. And so it gives you a pretty sizable, what's equivalent in humans to a middle cerebral artery infarct. My point here is just to show you how the infarct progresses over time, even with reperfusion. And that's something we also see in humans is, although we may get good reperfusion, we may also not get good outcomes and continue to have infarct. But my point is to show that at 24 hours, the infarct is histologically complete, meaning the infarct doesn't tend to grow. After 24 hours, the infarct is pretty stable. Lots of things are going on in that infarct, especially the immune stuff that we saw Marion talk about, but the infarct histologically is relatively mature. So what we did first is just take C57 black mice. They're all exactly the same genotype. They're in the same mouse house. They're eating the same chow. And all we did was separate them into either continued pair housing, which is PH, or into isolation. And you can see the isolated mouse has a much bigger stroke. It's the exact same stroke model. And you can see the infarct is 2 or 3 times larger in the animal that was isolated for 2 weeks prior to giving the stroke. But these animals also had very poor recovery and very big behavioral deficits. And you can say, well, that's not surprising because their stroke is so much bigger. So what we did to normalize the degree of stroke is we waited until 3 days after the stroke, when the stroke is histologically mature, then we separated them. And interestingly, we found no difference in infarct size. So in animals that were socially isolated, pair housed with a stroke partner or pair housed with a healthy partner, all the mice had exactly the same infarct size. But if you look at the survival curves, you see what happened to these mice over time. By day 30, almost the majority of stroke mice that were socially isolated, that's the line in purple, died. And they were not dying of their stroke. They just continued to succumb to the effect of the stroke they had a month earlier. And we didn't see that in the animals that were pair housed. And we then took our animals and looked at some of the biological basis for this. And one of the first things we did is a microRNA screen. And this was just showing that the males and females had a very different microRNA response to isolation. So we took some of the animals that we had separated 3 days after stroke. Again, their stroke is the same size. So we're not looking at microRNAs that are impacted by the stroke itself. We're looking at microRNAs that are impacted by the isolation after stroke. And what we found very interestingly in aged males that were isolated after stroke. So this is day 7. This is 4 days after they were isolated at day 3. A specific microRNA 10A5P came up as very robustly upregulated, but only in males. So then we said, okay, well, that's interesting, but does it mean anything? So we gave an antagomer to that microRNA, and we show in that first panel, you can see the pair-housed have very low 10A5P levels because they're pair-housed. When you socially isolate them, you see an increase in this microRNA, and we were able to inhibit that microRNA using an antagomer, and we found a reversal of the detrimental behaviors we saw with isolation, mostly depressive phenotypes, like in the tail suspension. And in the corner, you can see what happened in females. We gave this antagomer, but it had no effect whatsoever because this microRNA was not increased in females. So we treated both males and females with the antagomer, but it only had an effect in the males. So this is kind of a transition point where we kind of said, okay, and this is where we kind of integrated it into other work going on in our lab, which is really looking at peripheral inflammation and looking at the gut microbiome, which is the colony of bacteria that live within your gut. We know with aging and with stroke, you get significant dysbiosis, which is a decrease in microbial diversity and an increase in pathogenic pro-inflammatory bacteria. We've shown previously that dysbiosis occurs very rapidly after stroke. You get gut barrier breakdown, and that mucus barrier gets destroyed, and these pathogenic bacteria, which of course are much worse with aging, can cross the gut barrier, get into the bloodstream, and cause infections. So we decided to take a look and see what was going on with isolation. Just isolation. This is not stroke. So we had previously done fecal transfers of stroke biome into normal mice and saw tremendous amounts of inflammation. So we wanted to see, does isolation itself cause dysbiosis or something in the gut? And indeed it does. So you can see in the blue is the pair-housed animals, and in the red is the isolated animals. And we found that when we isolated the animals, again, we isolated them at day 3, and you can see that the isolated animals gained weight. These are not stroke animals. These are just regular uninjured animals. So when you take C57 mice and you separate them, the mice that are isolated gain weight. And when you look at their cecal weight, which is where there's a big abundance of microbiome, it was dramatically larger in the isolated mice. We found that social isolation did similar things to the gut that stroke does. It increased gut permeability and increased gut inflammation. And you can see that quantified on the left. And we saw a very big increase in gut cellular infiltration and in the blood as well. We also found metabolic phenotypes. So this ties in very nicely with the human data showing that isolated patients have higher rates of obesity and diabetes. These isolated mice, again, uninjured, these are actually, I should mention these mice are all middle-aged, 17 to 18-month mice. They became glucose intolerant. And the effects of the detrimental effects of isolation were seen very widely systemically. So we saw an increase in liver inflammation. We saw an increase in pancreatic inflammation. And we saw an increase in brain inflammation. So this here shows, just TMEM as an example, that the animals that were isolated had a reduction in TMEM, or the homeostatic marker on microglia, and they started having depressive-like phenotypes in the forced swim test, as well as in the tail suspension test. So we found that the animals that were chronically socially isolated had deficits in social behavior, including social interaction time with other mice. So isolating them changed their microglia, it changed their pancreas, it changed their liver, really far-reaching effects. I don't have time to go into it today, but doing whole genome shotgun sequencing and 16S sequencing identified a bacterial profile that was very pathogenic and was very notable for the animals that were isolated. So again, like Marion, both of us being neurologists as well as neuroscientists, of course, we wanted to see if any of this was relevant to our patient population. So doing a very small cohort, which we've subsequently enlarged, and we have much more data, but again, I don't have time to go into the nitty-gritty details today. But we took patients that were either interactive, meaning they weren't isolated, or isolated individuals. And they were, we used two different scales to measure this in humans. One is the Lubin scale, which is a social network scale. How many people do you talk to a day? That kind of stuff. And then the UCLA scale, which is a marker specifically for loneliness. Like, do you feel lonely? And they correlate very closely together, those two measures. But one is really more in isolation and one is really more of a loneliness scale. And we looked at their gut microbiome. We took dietary histories. We did metabolomics and transcriptomics in their blood. And we then did functional microbial analysis and looked at what the clinical impact was. So the Lubin network scales correlated with alterations in genes in our transcriptomic analysis that are related to insulin resistance and glycolysis, which probably explains why our mice were getting these metabolic phenotypes of obesity and glucose intolerance. We also found that loneliness altered a bunch of circulatory metabolic profiles as measured by metabolomics. So these social factors that we kind of didn't really pay that much attention to are really driving metabolic pathways and inflammation. We then took isolated individuals. I said we collected their biome, and this is a really small study that we're repeating now. We took fecal samples from the patients that were isolated versus the ones that were interactive and recolonized germ-free mice. So germ-free mice don't have a biome. So we took the human samples and recolonized germ-free mice with them. And we found when we first started that giving an isolated person's microbiome to a germ-free mouse actually caused them to die. So something incredibly detrimental in an isolated biome. We also have subsequently done this longer and have found that patients with that are isolated, these germ-free mice that get these fecal transfers also develop cognitive impairments. So moving a little bit into cognitive impairment, because I knew that Marion was going to talk about it, I wanted to talk a little bit about vascular cognitive impairment and dementia, which is, you know, as we heard about, very common, and with our aging population becoming increasingly common. And of course, we can see these diseases on MRI with subcortical infarcts, with white matter hyperintensities, cerebral microbleeds, and microhemorrhages. And one of the things we study in our lab in both animal and humans is cerebral amyloid angiopathy, which is kind of a classic mixed dementia, or where you can see both vascular disease. It really bridges both vascular disease and amyloid-related pathologies. And this is just where amyloid deposits in the cerebral vasculature, leading to microhemorrhages and in some cases macrohemorrhages. So this is one of our patients here. You can see the cortical microhemorrhages on the MRI as these little black dots. And unfortunately, this woman actually had a large lobar hemorrhage. We have a brain bank here at UT. And she and her family had asked to be involved in a brain donation. Here's her brain, and we can make the diagnosis of cerebral amyloid based on neuropathological imaging. So this is the same patient, and here's her hemorrhage. So what we wanted to now kind of do is pull together social isolation with microglial dysfunction, neuroinflammation, and some of these vascular patterns. So what we decided to do is now combine social isolation in a model of cerebral amyloid angiopathy. We use Dutch Swiss Iowa mice, which are, an okay model for CAA. And we isolated those animals for 3 months, and we found that these are both males and females. And you could see that after short social isolation, 3 months of social isolation, they didn't really have any motor deficits, but they started developing, and interestingly, the females developed it much earlier than the males, depressive life phenotypes, as well as cognitive deficits, as noted in the tail suspension, in the novel object. If you leave these animals in isolation for 6 months, both the males and females, the mice that are isolated get depressive phenotypes and get progressively worse dementia than animals that were pair housed. And so trying to tie this into our inflammation, as we had seen in some of our non-injured mice, we started looking at some of, first of all, does isolation cause amyloid progression? And the answer is yes. And does it cause activation of microglia? And the answer is yes. So you can see these are just happen to be the male ones, but you can see there's an increase in microglial activation, and that's measured by CD68. And what we also found is that microglia that's activated. We have some spatial transcriptomics data, but I don't have too much time to talk about it. But the microglia were very activated and phagocytic. And what were they eating? They weren't eating the amyloid, they were eating the white matter. So we can see here that the oligodendrocytes were really kind of the ones that took the brunt of some of the detrimental effects of isolation. So we think when you isolate an animal, you get this peripheral response as measured by that dysbiosis. You get a metabolic phenotype, but you also get a lot of things going on in the brain, including microglial activation, which leads to white matter destruction. And you can see here, here's a activated microglia eating some white matter. And you can see CLEC7A, which is a kind of a typical marker of, a very activated specific state of microglia increased with social isolation. And again, these animals didn't have any injury. They were CAA model mice, and they had a lot of phagocytosis of white matter. So this is a very busy slide, but it's my last slide about how we're kind of trying to link social isolation to peripheral inflammation and microbiota changes with central inflammation. So this may be one of the ways that the brain and the periphery communicate to activate immune signaling as well as neuroinflammation. So I just wanted to also give a shout out to our group, because we work as a very large group here at UT called the Brains Research Group, and every single person on the slide has contributed something to one of our kind of myriad of different programs. So I'll stop there so we have some time for questions.
Thank you so much, Dr. Buckwalter. Very great talk. Now let's open the discussion. Please feel free to type the questions in the Q&A. I start to see some questions. So the first question is for Dr. Buckwalter. It's from Lukman. Are there any trials going on for anti-VCAM and anti-VRA4 for post-stroke dementia patients?
That's a great question. The answer is no, although I think a trial would be very exciting. There's currently no anti-VCAM antibody that works against human VCAM. The antibody used binds the mouse, but not the human. But for anti-VLA4, there are actually 2 FDA-approved antibodies that are used in autoimmune disease, in MS, and in Crohn's disease. So we're looking into ways to get a trial funded. Dementia trials are very expensive. And we're also working on the best ways to use the biomarker to identify people with blood-brain barrier breakdown. So hopefully in a few years, we'll have an answer for you on whether they work, but we need money first. So we'll see.
Thank you. And the next question is for Dr. McCullough. Some of the effects of socialization can be reversible.
Yes, that's a great point because they can be. And again, this is where it gets a little subtle because some people may be isolated, but may not want contact. And giving them that contact may be more stressful than leaving them alone. So, yes, there's been a lot of new studies really coming out to really enhance patient kind of support. And I think those will really tell us a lot about whether we can reverse some of the detrimental effects of isolation and loneliness. How do we engage those patients? And then how do we model it in the lab? In the lab, we can re-put the animals in together. It's difficult with males because if you separate males for too long and put them back together, they fight. They can be very aggressive. So we are doing those experiments now to kind of figure out how tractable is this? Is it something that we can reverse? And I think the answer is going to be yes. And I think we'll be able to reverse it by giving caregiver support and patient support. But again, it can be very expensive to think about giving a social intervention.
Thank you, Dr. McCullough. And the next question is for Dr. Buckwalter. Um, have you checked another pathway important for parasite biology in your studies? And did you see anything related to the smooth muscle cell compared to the parasite? Thank you.
Okay, great question. A lot of the antibodies that we used were actually antibodies that mark both vascular smooth muscle cells and pericytes, which are closely related cells, and they both respond similarly to PDGFB. So, although we couldn't distinguish between the two in our human samples, we just didn't have enough tissue sections to do it. It looks like both are affected. In the mice, it seems like it's more the distal pericytes. And lots of work remains to be done on this. This is kind of hot off the presses data. So I think that would be a really great question to ask going forward.
Okay, thank you. Next question is, does the steroid help improve outcomes in the post-stroke dementia patients?
I guess that's for me too.
Yes.
Yeah, like I said, this is really new research. And so to find out whether steroids would help these patients, you would have to do a very large, expensive trial. I think it wouldn't be the first trial that I would start because we know that long-term use of steroids has a lot of side effects, including increasing the risk of diabetes and other cardiovascular risks. And so I would worry that patients who already had a stroke and have cardiovascular risk would be at high risk of more side effects than benefits from steroids. But it still could be that they would help. I think this is sort of a juncture where now we have this new discovery and we have to pick and choose which things we're going to test. But I would favor better tolerated immunomodulatory therapies than steroids.
And I have a question too for you, Marion. Do you know if they've done any follow-up in the patients like treated with like rituximab or I mean, again,
Yeah.
it's confounded because they have MS.
Yeah.
But you know, those chronic immune treated patients, do they have a lower incidence of dementia?
There is a little bit of data on natalizumab and cognition in MS, and it does seem like long-term treatment with natalizumab, which is anti-VLA4, does improve processing speed and cognition in MS patients. I would say
Compared
that
to somebody treated with a different immune modulator.
no, compared to their historical baseline. And what I'm
Okay.
about to say is these are moderately small studies. Most of the studies with MS don't focus on cognition. So you can take it with a grain of salt, but I think it's kind of promising that it may be improving cognition. And it does really improve quality of life and long-term outcomes in people with MS. Rituximab, I'm not aware of any studies with. And again, it would be very interesting to know.
Okay. So The next question is for Dr. McCullough. With the gut microbiome theory, can specific diet can reduce the effect of loneliness?
Yes, that's a great question because I didn't get a chance to talk about it. We've been looking at this for like 10 years. So we have tremendous amount of stuff on stroke and aging and age-related dysbiosis that really show that there's certain bacterial strains that are very detrimental. There are certain bacteria, and it may not be the bacteria that are important. It's probably more important what the bacteria make. So some of the more healthy anti-inflammatory metabolites that a healthy biome makes are short chain fatty acids and indole metabolites. You may have heard of the detrimental one, also made by the biome, is TMAO, which is a really kind of potent oxidant, damages the vasculature, linked to heart attacks, linked to stroke, and that's made by detrimental microbiota. So one of the things that the microbiota do is they make stuff. They make these metabolites. What do they use? They need a substrate. So if you give them a healthy substrate, meaning diet, you can change the microbiota composition and you can change the metabolome. And the best one so far that seems to be coming out in the literature is really a Mediterranean diet. These healthy microbiota really like fiber. So a high fiber diet seems to be the most effective at reducing inflammation. So that's the Mediterranean diet. Lots of, whole foods, little processed foods, low red meat seems to be the thing that shifts your biome to make these healthy metabolites.
Thank you. So next question is for Dr. Buckwalter. What are the difference between stroke subjects who had a BBB leakage than the other one who didn't?
So, so far, we're just looking cross-sectionally, and the people with BBB leakage tend to have more hypertension, which is perhaps not a surprise. But we didn't see any other differences in the groups so far. And the data I showed you was from our first 56 participants, and we're hoping to get up like 150, 200 participants with these scans. So as we get more people in the study, we may be able to see other things that are different about the people with BBB leakage versus the people without.
Mm-hmm. So basically, what is causing that chronic leakage in patients?
Yeah, I mean, I'll just put a plug in. The best data for preventing dementia is Mediterranean diet, like Dr.
Exactly.
Pollack just said.
The biome
And it wouldn't
steering.
surprise me. It wouldn't surprise me if all of the things we tell our patients about get your cardiovascular risk factors treated, have a healthy diet, get enough sleep, Life's Simple 8, which I think you can find on the AHA website. I bet you all of those things contribute, but we have to prove that, obviously.
And it's interesting because it's the same as gut barrier, brain barrier, gut barrier, and these are very kind of
Yes.
metabolomically regulated.
Mm-hmm. Yeah, I was going to say, combining your talk, what do you think about the framework of from social isolation to gut microbiome and then blood-brain barrier and then post-stroke dementia?
It all
Right?
ties together. I think,
I
Yeah.
think, yeah, agree.
Marion, there's a grant in there. There's a PPG. I sense
Here
it.
we go, Louise. I think stress is really interesting as a risk factor. And Louise mentioned that we had sort of communicated by text about what she should talk about. And I think the social isolation is really fascinating. And it really tells you that what you experience really matters, like taking care of yourself, making friends, having social connections. It really has a biological effect.
And that's why I like the fact that you can
Very,
model it in animals because
yeah.
it's not even so soft in a way. This is biology. It happens in a
Yeah.
mouse.
Yeah, and I think it'd be really interesting to look at in some of our post-stroke cognitive decline models, as well as in our patients.
Okay, so in the interest of time, we'll take the last question. It's stroke rehabilitation in humans often involves social enrichment activities similar to what we might do in post-stroke murine models. Has any work been done on these immune models comparing stroke rehabilitation interventions?
And Marion, you might know a little bit about this. There hasn't been anything really dramatic. We're still finding that a tough nut to crack, right? Like there have been trials even in the past year looking at, new therapies and transcranial stimulation, and again, trying to really find the best things that you can do to help stroke recovery. Certainly having a good caregiver network, a good social network is going to be part of it. And I do think that if we can find what's detrimental in social isolation, then we can target that and try and also figure out what enhances with social integration. And I think that's got to be a really important part of rehab. And, again, in animals, it's easier to model because in humans, you could say, oh, well, if somebody doesn't have a caregiver that can take them to outpatient therapy, they're not going to get therapy. But I think when you think about things like using tele-rehab and stuff, I think we have to think about, okay, how can we optimize these for interaction with other people?
Okay, so thank you again for our speakers for a wonderful talk and discussion. Thank you everyone for joining today's webinar. We hope this session sparks new ideas and collaboration across the stroke research community. And when the webinar ends, a short survey will pop up. We would really appreciate your feedback. And thank you again for joining us. We look forward to seeing you at the future sessions.
Thanks, Hanson.
Thank you.
Thank you.
Bye.
Bye, everybody.